Watson and Crick said in their famous paper reporting the discovery of the structure of DNA that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So what exactly did they mean by this? We'll take a look at how DNA replicates, or makes a copy of itself. By the end of this lecture, you should be able to describe the process of DNA replication and compare and contrast the synthesis of the leading and lagging strand. Replication on a DNA molecule begins at places called origins of replication. These can take one of two forms. They might look like forks. Replication forks are Y-shaped structures where DNA separates its two strands and adds new bases to the original parent strands. Replication bubbles can also form. This is where replication begins in the middle of a DNA strand as opposed to the ends, like you would see with a replication fork. Eukaryotes, or any organism with a nucleus, has hundreds of replication bubbles as it synthesizes new DNA during the S phase of the cell cycle. With billions of base pairs, many bubbles are needed so that replication can take place simultaneously at many places on the DNA strand. This ensures speedy replication. Bacteria have just one replication fork. They have much less DNA to copy. There are three main steps to DNA replication. The first step is that strands of DNA need to be separated. This is done by the enzyme called helicase. Helicase goes through the DNA and breaks the hydrogen bonds between nitrogen bases. In order to prevent them from rebinding to their partners, binding proteins, single-stranded binding proteins, attach to the DNA so the helix remains unwound. The second step is priming. In order to mark where replication should begin, small, single-stranded RNA molecules attach to the DNA to signal cells where new bases should be added. These small pieces of RNA are called primers. The enzyme primase creates these short RNA primers. The next step is the actual replication of the DNA. An enzyme called DNA polymerase attaches itself to the RNA primer and begins to attach free nucleotides in the 5' to 3' direction. DNA polymerase doesn't just add any nucleotides as it travels down the DNA. It attaches complementary nucleotides. So where there's an A in the original parent strand, a T is added to the newly synthesized DNA strand. C's are added to G's, and vice versa. DNA polymerase runs into a small problem. It can only add nucleotides in the 5' to 3' direction. But because DNA is anti-parallel and both strands need to be copied in order to have two complete strands of DNA, in the end, DNA polymerase can only synthesize one of these strands continuously. If you look in the small picture in the corner, you'll see what I mean. So imagine DNA as sort of like a zipper. Helicase is continuously unzipping DNA so that in this picture the replication fork moves further and further up. You can see the newly synthesized green DNA can essentially follow the helicase up the molecule with no problem. However, on the other strand, 5' is nearest to the replication fork. So nucleotides must be added in a manner such that the DNA polymerase is moving away from the replication fork. As helicase continues to unzip, DNA polymerase must jump up the replication fork and synthesize a short fragment of DNA until it reaches the already synthesized DNA. By the time it gets there, helicase has unwound more DNA. So DNA polymerase must unbind from the DNA strand and move back up to where the replication fork has moved up. The strand that can be synthesized continuously is called the leading strand. The strand that has to be synthesized in small parts is called the lagging strand. This is because it's always trying to play catch up. In this picture, imagine the replication fork is on the left side of the screen. The bottom strand of DNA is made of new bases, so there's no problem adding bases to in the 5' to 3' direction. On the lagging strand, DNA polymerase starts at the replication fork and moves 5' to 3', but as it's doing this, more DNA is becoming separated behind it, so it needs to go back and start over near the replication fork. This process will continue until all of the DNA has been copied. The series of short fragments this creates on the lagging strand are called Okazaki fragments. The last step in DNA replication is that all these Okazaki fragments must be connected together. DNA ligase does this job. The replication of DNA is considered semi-conservative because each new strand of DNA contains one old strand, or parental DNA, and the other strand is newly synthesized. In the picture, the original strand of DNA is red. The second picture shows hydrogen bonds being broken and new bases being added to the old existing strand. The final picture shows the results of DNA replication. 2 new DNA molecules, each one with a parental strand and a newly synthesized strand. In your notes, create a table showing the enzymes used in DNA replication and their functions. What would be the complementary strand of DNA for the following DNA sequence? Write down your answer, because here comes the correct answer. Now that you're done, don't forget to go to the assignments folder, click on the DNA replication quiz, and complete that by midnight tonight. So here's a preview of the questions.